First things first, I'd like to get my slides set up. And I need to close that view. Okay, let us take a brief moment to acknowledge the land where the State Water Resources Control Board offices are located. Sacramento was and is still a gathering place for many local tribes who have lived throughout the Central Valley and the foothills for generations. They are the original stewards of this land. These people include the Anishinaabe, Maidu, Miwok peoples, and the Patwin and Wintun tribes. They have remained committed to the stewardship of this land over many centuries. It is our hope that if we acknowledge that our organization sits on the lands of Sacramento's indigenous people, we recognize that we are here because of the genocide, slavery, and sacrifices forced upon them. And in remembering these communities, we honor their legacy, their lives, and their descendants. Excuse me. So I also want to thank you all for joining us. We've got a great cohort this year. We've had, since our last meeting, we've had some leave and some enter. Our newest being Preserve Calavera down south. So that's pretty exciting. We were happy that one guy dropped out and we were able to pull them right in. So that's great. And thank you all for your participation and making this thing happen. We really appreciate it. What I aim to do today is take you through everything we could think of about collecting your samples. So without further ado, I'm gonna just shift my screen a little bit here, make it easier for me, and get right into it. So when to sample? If you're looking for a certain species, let's say a certain salmonid, and that's really what you wanted to use these kits for, you're going to want to do it when the target species is most likely to be in the water body. So, you know, if spawning season is a good time, that's probably a good time.
Hey Peter,
you know,
are you sharing slides? Are you sharing slides with these? Okay, we're only seeing the SeMMAP cover page here.
Oh, thank you.
There you go.
Oh yeah.
Yep, that works.
Okay. Thanks, Charles. So yeah, to coincide with regular monitoring, you know, that's most of what we're doing. That's certainly what our friends at the California Department of Fish and Wildlife are doing with their study to compare against traditional methods. So they're taking their DNA sample first and then going in there and collecting their bioassessment data. And that's a great way to do it, because the best use of these kits is to look at biodiversity and everything that's going on. But we're not dictating how you use them. If you want to search for one species, you can do that, but then you're sort of leaving out this whole bigger picture of everything that's going on in the water. The other things to consider are sampling just during regular flows, typical flows, not too high and not too low. Some things like in late summer, higher bacterial and algal loads may interfere with, they may clog up your filter faster. And you also want to consider any migratory patterns of species. Okay. So I made a little watershed here, just an example watershed. Can you see that slide, Charles?
Yes, sir.
Okay, great. So this is just an example. And if you were to do biological monitoring and you had 8 kits, the best way to go about this would be taking duplicates and probably at the points along your watershed where you're getting the most influence. Here I did the pour point, you know, the end of the catchment there, where this long stream comes in, taking a sample there, and where this other long stream comes in. So something like this would be perfect. The other way you could do it is using a reference site. A reference site is what is most indicative of untouched natural state of your waterway. There's more direction on reference sites in the EPA documentation, and I can get that to you if you're interested in that. But this is basically our reference. This is the untouched look, and then we can randomly sample in here and of course at the pour point to sort of measure against what should be and the results we're getting along the way. You can also think in terms of seasonal monitoring. In this case, April, June, August, those summer months when water is flowing, but that's only for my pretend example watershed. If you have more water flowing in the winter, you know, do it according to what you think is best. You know your waterways better. But again, in this example, we're taking duplicates because duplicates are going to be your best way to get a good snapshot of what's going in the water to get the most high quality data. So are there any questions at this point when we consider sort of the larger landscape, you know, where to questions?
And just to clarify, everyone, these are suggestions is really what they are. I mean, in a typical use scenario, what you are interested may differ. So that's why we asked submit, you know, a small kind of project paragraph of what you're interested in, so we can make sure that we touch base with you and ensure you guys are getting the most out of these kits, and they're going to answer the questions to the best of their ability for your water, you know, your watershed and your questions. What are you looking for exactly? So there's a lot of different ways to sample, especially, you know, geographically. So if you do have any more questions, or if you think, you know, you want to refine the way you're going to sample, please, you know, send us an email. We can set up a chat and make sure that you're getting the most out of them if these slides don't answer your questions.
Absolutely. And if you want help, like I can help you with your watershed. I'm a whiz with maps. So anything like that, hit me up. We have a lot of great resources at the Water Board and a lot of great data to look up that may help you with your decision making. So moving on with the where to sample question, now we're talking about more in the waterway, and we want to avoid stirring things up when we enter the water prior to sampling and maybe getting DNA that might be on us into the mix of what you're trying to collect. Always stay downstream of your sampling points, and consider anything, you know, any human influence, sewage pipes in this instance, and close human dwelling houses and stuff like that. You want to collect your samples in areas of high mixing, maybe not rapids, but a good riffle kind of thing, and high mixing and high visibility. You want to stay away from sort of muddy spots that might clog up your filter too quickly. The other thing to remember is to sample regular intervals along the river network, depending on what you're doing, and to consider tributaries and any connected water bodies that might influence the samples, kind of like the things I was showing you in the watershed example. The other thing, please be consistent about how you sample, and please document how you did it. There's notes in the Jonah DNA app and notes in the Survey 123 app for you to journal how you went about, how you decided to sample where you sampled and how you did it. If you took samples across, from bank to bank, write that down. It's going to help you out next year when you go to sample again, and it's going to help science in the long run. So please remember that. Whoops. How many samples should you take? And this is a big question that we get asked a lot. If you're looking for a good snapshot of biodiversity, take duplicates. I would say in most instances, take dupes. This is another thing you want to write down and make sure to document. In the Survey 123 app, it's going to ask you if this is a single or a dupe. I see we have a question in the chat. I'll get back to you on that, Alex. The number of samples, if you're looking for an invasive species or an elusive species, there might be a better way to go about it than taking duplicates. But because metabarcoding looks at populations, that's really sort of the best case scenario, the best uses of these kits. I'm not saying you have to do that, but you'd sort of be missing out if you went another direction with that. But absolutely can, if you want to look for cyanobacteria, wait till those late summer months and see what you get. And we can definitely help set that up for you and give you a good idea of what that sampling looks like. And you may want to just consider distinct subhabitats, maybe pools or areas of different flora vegetation, depending on where your species of interest may like to hang out. So again, some replication at each site is always recommended to increase confidence in the data. Other things to consider, the volume of water can really vary widely. These things clog up for what looks like no reason. You'll have the same clear water you had just downstream, and it'll clog up pretty easily. Factors that may affect this include turbidity and sediment churning. But we've been averaging somewhere between about 180 mil, which is 3 syringes, and 500 mil, which is that's pretty high. But I remember we've gotten close to 8 on one. It just never stopped flowing. I don't know if we had a hole in the filter or what was going on. So eDNA distribution in river systems is not always the same. It can be very, very dilute compared to lentic samples. Again, just document your methods and decisions to help from year to year and to help us out. Take consistent samples, just do it the same way, call things the same things, that kind of thing. And take care not to disturb the stream bed before sampling. So were there any questions about those suggestions and those considerations? Okay, we will move on. We made a new collection video for you guys, and we will be watching it now. Let's take a quick look at the contents of the kit. Here are your gloves to keep things clean. This is your collection syringe. This end here screws onto your filter housing. The small syringe is the preservative syringe that you'll put in at the end of collection. This is your filter housing. It's contained in a black bag that is sealed to keep out contamination. When you're done filtering and putting the preservative into the filter housing, you'll put these caps on here on both ends of the filter housing and put it right back into the black bag to keep out any more contamination. The black bag also contains the QR code. You'll use this code to enter into the Jonah DNA app. And lastly, you should, with your kit, have a self-addressed stamped envelope to head out to the lab.
So I'm going to go ahead and take the filter off. I'm going to just rinse my gloves downstream of where we're collecting the sample a little bit. That way, if there's any residual anything on my gloves that can interfere with the sample, it's not there. And I'm going to do the same with the syringe as well, since that's the part that we're actually putting into the water to collect the sample. So I just want to make sure anything that's on me that could potentially contaminate the sample is washed away. Keeping the filter out of the water. Our first 60 mil. And then again, the smaller end of the filter onto the syringe. I would let the air bubble come up to the top so that the air is the first thing coming through the filter. And then you don't want to use too much pressure when you do this because you have the possibility of damaging the filter. So you want a steady stream, but don't force the water out. Already I'm getting some resistance at the top of the filter. Try to dispense your water downstream from where you're actually taking your sample a little bit, just so that you're not potentially having mixing between your filtered water and the water from the stream. I totally plunged through that first 60 mils, so we take our filter back off. I'm just going to repeat this process until you can no longer push more fluid through. So now it's going to fit again. We put the smaller end into the screw end. We're going to seat our filter with preservative. You use 1 mil for this whole syringe, it looks like.
If you can, I think there's usually leftover.
Is it ethanol? Like, am I told to just discharge it? Or?
Yes.
Okay.
Put your cap on.
So put our first cap on the one side. I'm going to take our second cap. So now this one, you have the wider end. So it should look like that. Now that goes straight back into the black bag where the filter came out of, and we're all done. Let's see what else. You're good to go.
right, so there's our latest video, and we'll share a link to that a little later on. So now I'd like to talk about, were there any questions about that after watching that? That all made sense? I'm good. Yeah.
Oh, I was wondering, how hard should you push onto the syringe to find out like the filter's clogged?
If you use 2 thumbs to start pushing on, like Lindsay was just using 1, but you'll eventually have to probably go to 2 thumbs to get, as long as it's flowing through there at a decent rate. Once it gets down to like you're pushing and you're only getting drops, you're done.
Okay. So
Cool.
no more than 2 thumbs, you're saying?
Yeah.
Don't palm
it. Yeah. You'll feel, I mean, you'll feel it. And certainly, you know, this was Lindsay's first time doing it, and you can see where sort of fiddling around with these sealed bits and pieces gets a little bit fiddly, for lack of a better word. But that comes with practice. You'll sort of get your groove on probably right around kit number 7 or 8. So yeah, were there any other questions?
I have a question. What is the purpose of pushing the air through at the end?
Just drying off the filter a little bit so it can accept the preservative.
Okay, thank you.
When you dry it off and introduce a little bit of ethanol, you kind of halt, because you could have actual microorganisms in there as well, which can actively degrade the sample. So drying it out reduces the water activity, introduces ethanol as a preservative, and lets them keep longer.
Thank you. Perfect.
Can I just add to the question about the filter? Because I mentioned that during the video. You can definitely apply a little bit of pressure before you're actually going to rip it. So I got to a point where I was just using both my thumbs, and it was pretty hard to push through. It's just like a best judgment kind of situation, because if you really are forcing it, you could possibly create a tear. But it's not so delicate that it's just gonna, like, you know, rip right away if you put pressure on it. So I didn't want to create that idea or anything like that.
And if it does tear, it's not the end of the world, right? It would hinder further sample processing for that specific filter, but there'll still be, you know, product on throughout the filter that they'll be able to extract. So it's not the end of the world. It's just, you know, when you go to push the preservative in there as well, it could have a larger pore opening, so you don't get good like mix of the preservative throughout there. So not the end of the world, just not great.
Anything else? Okay, after you take your sample, you will want to enter your results in the 2 phone apps. And this is, you know, I wish we didn't have to do 2, but one connects to the lab and one connects to us. So that lab data is going to the lab, and both of the, you know, the unique ID that's given to you with your Jonah kit links your data to the lab and to us. So it's real important to get that one right. And getting the phone app is really easy. Just go to your app store and download the free Jonah DNA app. It's both in the Apple and Google stores, and easy enough to download. I'm going to send you all an email with the usernames and passwords for this to the SeMMAP partners. I just, you know, sort of want to keep that stuff as private as we can, so we're getting good results. So I'll mail you written directions, links to we have a video, a video for the apps, and those passwords and stuff, I'll email them to you shortly after this meeting. So you'll download, input your special email address and password, and then it will ask you for the kit ID. There's two ways to enter this. You can enter it by hand or using the QR code that's on the kit. It's 50-50. I've had luck scanning the QR code, but sometimes never. Next thing, it will ask you for site name. This is up to you, but this is where consistency comes in. This is going to become the official site name in our database. So please be sure to write it consistently from now on. So we're going to create a unique station code that ties this information to help with our data management. So for example, I mean, I think most of you probably already have site names for your site, but if you don't, you know, it'd be something like Minnow Shores #1. Just something simple like that is good enough. And if you're taking duplicates, just say Minnow Shores 1 duplicate for your second sample. If you don't, it's not the end of the world. We get that information through survey 1, 2, 3, but it's nice to double up and have backup on that. You'll enter your sample type, which is aquatic, and the volume of water you push through, so 60 times however many times you were able to pump through. Hit the little compass down here, and it will automatically grab your Latin long. And by default, the time is the time you are entering your data, but you can change that if you need to enter this information later on down the line. So then the only thing else it's going to ask you is if you want to make the data public. The default is no. Yes means you can share it with Jonah DNA, and they have a very similar dashboard to what we have on the SeMMAP Data Resource Hub, and they will share it there. So that's up to you. Do whatever you'd like. And there's a notes section to just document whatever, you know, if you drop the filter or something weird happened, just make a note of it and hit the submit button. It'll give you a choice to submit now or save it for later when you get back to somewhere where you have cell service. So pretty simple on that one. Ours is a little more robust. Are there any questions about that? That one's pretty easy, really. The Survey 123 app, you'll go to the App Store and download ArcGIS Survey 123. It'll ask you to sign in, and this again will be on the information that I send you. It's got a separate username and password for that. So you'll enter those and sign in. Once you've signed in, it's going to ask you to download surveys. This will keep the survey on your phone so that when you're out of data range, you'll still have access to it. So that's what it looks like with this. I don't know why it squashed the circle, but it did, and that's what it likes to do. So it stays. So once you've downloaded that, you'll hit the back button, and that will take you to your downloaded survey. So when you're out in the field, this is what's going to appear when you open up the app. So you'll click on the survey, and I need to share a new screen with you guys, so give me 1 second. There we go. So this is what the SeMMAP eDNA field measurement form looks like on your phone. And one of the neat things, when we inventoried all of your kits, now you can open it up and we'll select Sierra Streams because we like Sierra Streams Institute. And now we have all the kits that they got, and you'll just choose your kit from this. And if something went wrong, you can hit other and input whatever the kit is and just hit enter, and that'll record it. Again, by default, the date and time are already filled in for the date and time you're doing this. For the collections site, let's see if I can do it on here. I don't think it's going to allow me to, just because I'm on my desktop. But basically they have another compass icon, and that will automatically put in your let and longs. You'll check those against what you're seeing on the map. And this is actually just slightly off from me. So I can zoom into the map, pick that up by clicking on it, and drop it over there, and I'm good to roll. You can also take photographs. It'll take 2 photographs or up to 10 megabytes. You can use photographs that are already on your phone, or just shoot straight from your camera. I'm going to answer one question from Christine. If we do not share publicly with Jonah DNA, will our data still be included publicly with the SeMMAP Data Hub? And yes, that is what will happen. So you won't be on the Jonah DNA dashboard, you'll just be on ours. So moving through, the next one is the SWAMP station code. Most of you won't have these. Our regional board members and CDFW usually have these station codes. We'll make one for you, so just leave it blank. Site name, remember, we're consistent with whatever we're calling it on the Jonah app. Sample number, this is going to ask you if this is the first of 2 samples. So if you're doing dupes, we want to know, or we're only collecting 1 sample at this site. So just punch that in. Anything with a red asterisk is you have to fill it out. Most of these things have something for like not recorded or something like that, but others, you're going to have to answer one way or another. So location and waterway, pretty easy. Bank, mid-channel, open water. If you're saying mid-channel and what you really did was sort of go bank to bank and take 4 syringes from bank to bank, just make a note of it. That we just want to know. Moving on, dominant substrate. A lot of these are pretty self-explanatory. So we'll say it's gravel. We took it in mid-channel. Water clarity, it was very clear. Observed flow. Gee, I don't know how to do this by just looking at it, so I'll just say water flowing. Then we'll enter our information for temperature, pH, specific conductivity, dissolved oxygen, turbidity, flow if you have it. If you're using a flow number, flow meter, it'd be great to have this information. Or if you're sampling from a site with a gauge, it'd be really great to get that information, but not required, because we know a lot of folks don't. So the dominant land use, again, pretty self-explanatory. We'll say agriculture. Sky code is kind of a weird one. This is a swamp specific one, but all it means is what the sky is like right above where you're sampling. It was a clear day, so that's great. Animal present, when we were out, we saw cattle and bunches of birds, so birds and domestics. No precipitation within the last 24 hours. No evidence of fire. And then another oddball with Beaufort scale, it's how's the wind outside. And what we need for this is a number. So it kind of looks weird here. The numbers correspond to whatever the description is, and these are the official Beaufort scale descriptors: fresh breeze plus, and fresh breeze plus plus. So again, you can put not recorded on here, and that's perfectly fine, but we'll just say there was a moderate breeze that day. And we have notes to, document how and why we did what we did. You'll hit the submit button. Oh, and it told me I forgot something, so I will enter that. And this is a required question. Now we'll see if we can submit. The submit button doesn't work in preview mode. Great. It's going to ask you whether you want to submit now if you're within data range, if you want to save it to your outbox, if you want to wait until you get back to the office or somewhere within range. And it will also ask you to just save as a draft, and you can go back when you get back to the office and maybe make some changes you wanted to make or something like that before you submit. So there's sort of a lot on it, but again, you'll get a groove. They're pretty self-explanatory questions. Is there any questions about how to operate any of this or what we might be looking for?
I have a question.
Mm-hmm.
Okay, for the time, we don't usually do turbidity in the field. We do that in the lab. So then when we submit, we need to correct the time for the actual time we took the sample. Is that correct?
Yes, but if you save as a draft, it will save when you took that sample.
Okay. Okay. So then we can just put in the turbidity number when we get that in the lab. Okay. Thank you.
Yep. Anybody else? There's got to be somebody.
Yeah, if you'll allow me to comment, your lab turbidity rating is going to be different than the one you take in the field, and you probably should note the time.
In the notes. Good point, Bud. And Sarah asks, when do you need the samples collected by? It's whenever you have I mean, if you are going to wait, like if you're down south and your stream's not running, you can wait till the fall to take it. Not a problem, but sometime this year would be nice. Oh, and the other thing I didn't mention is once you're done taking the sample and you put your filter into the black bag and then into the blue envelope, you can put as many filters in that blue envelope as you want to, as will fit comfortably for mailing. So if you just want to save on mailers for the lab, feel free to put 2, 3, or 4 filters in the same one. They can the only thing we ask is that don't expose them to high heat or UV radiation. So don't leave them on the front seat of the car and then go to the grocery store. But they will, they're pretty robust. They'll last under a lot of circumstances, but the sooner you send them in, of course, the sooner you get your results. And we're looking at like 4 to 6 week turnaround times. So just thoughts. Sarah asks.
Can I ask, just clarify what he said about the turbidity? So in the notes, I should clarify that turbidity was taken in the lab at a different time and record the time. Okay, thank you.
Yes. Sarah asked how many kits came to each of us. Most got 8 kits. I think just about everybody, but the Department of Fish and Wildlife, we're engaged with a larger study with them, so they actually got like 60. But that's because we wanted a really consistent, robust data set that compares to what they're doing with traditional bioassessment. So, but otherwise, I think everybody got 8, maybe some got a little bit more, a couple of more, because they were doing some other work, but 8 is pretty much the base. Any other questions?
So this is also a great time to remind everybody, if you haven't submitted a project kind of paragraph or just kind of an outline of what you're interested in, what your study is, how you're interested in using these kits, please do so. It kind of helps us keep track of where they are. And again, we want to make sure that you're getting the best use of this data as possible. Maybe what we might do next year is if we get funding for more kits, we ask you to submit those, and then we can try to make sure you have enough kits to do the monitoring, based on the previous year's data, something like that. So please, if you haven't done so, please reach out to us with just a general outline.
And this is Rich Vanderson, Region 1. I was just wondering about the cyanobacteria. You mentioned that a time or two. Is cyanobacteria part of the eDNA collection and processing?
Yes. So we'll generate all phytoplankton across the board. And I have a reference list of all the different organisms that they have as part of their database that I can share with you as well.
If you could, that'd be great. I appreciate it.
Yeah.
Yeah, I have a quick question. This is Nathan Mack with Fish and Wildlife. I already did some collecting like a week or two ago, and I mailed in the kit and I entered the data into my app, but I didn't use your sound like you're sending out usernames and passwords to people. As long as I marked it as public data, is that something you're still going to be obtaining through Jonah, or do we need to go back and make sure that tracks back to you? And then also, can I continue to use my Jonah account on my phone as is?
Yes.
Okay.
Are you, Nathan, you're the only, Sean's crew isn't taking these kits, right?
It's just me.
Okay. As long as it's consistent.
Okay.
I think that's fine.
It'll stay the same, username as what I used for everything last year.
Okay. Yeah, then I can get that, because as long as we're sharing that, we're good.
Okay, good.
Cool. Thanks. I'm glad you brought that up. Oh yes, I can. Crystal asked if I wanted to if I could send out the organisms that will be analyzed so she could help with her study design. Yes, I can do that. Absolutely, Crystal. And also let you guys know that that is available on the SeMMAP Data Resource Hub. It's on the 3rd page. It's under collection, I think, is the header, eDNA collection. It's down there and it just says taxa lists, and you just click on whichever you're looking for, phytoplankton, macroinvertebrates, or fish. And it'll link you right to those lists. But I'll send it out to you, Crystal, for sure.
So I just dropped a link to that page in the chat. And if you scroll all the way to the bottom, there is 3 different buttons for macroinvertebrates, fish, and the phytoplankton taxa list included there.
Thanks, Charles. Anybody else? That is all I've got to share. I just wanted to make sure you guys had everything you needed to know before heading out and taking samples. I suggest downloading and saving the information I send to you as soon as possible. You just don't want to get out there and realize you forgot something or don't have the passwords you need. It's not the end of the world if you don't, because you can always fill it out when you get back. Oh, and the other thing I wanted to mention is we also have paper forms if you want paper for backup. If you're not using your own, we have those for that. I will put, yeah.
I got a couple comments here. I checked on the partners page for Blue Lake Rancheria, and some of that information is kind of confused with some other tribal information. So some of that stuff's kind of mixed around, and it keeps asking me for a sign in whenever I hit that link. So I'm not sure what's going on that end of things.
Ooh, can I hit you up later and we can do a little Teams chat and go over it? I haven't looked at it in a while, so
yeah, that's fine.
I
I can handle that via email, and I took a sample on the 23rd of March for Ulicon presence in the lower Mad, and I used my own. This was before all these instructions got sent out, so I'm going to try to back engineer some of these other instructions to match up with that. So I guess I'll just follow up with you via email on the best way to do those things.
Yeah. We'll figure it. We'll get that sorted out.
All right, thank you.
Before you guys go, I want to make sure that you have all the links that Karen asked for. I just need to copy them and include them in the chat. Really? That's not fair. It's not letting me paste. There it goes. There's links to absolutely everything in there. And if nobody has any further questions, we are done for the day, unless you had anything you wanted to add, Charles.
Nope, I'm good, but please reach out if anyone has any questions or needs any clarification about how study design, or if any of the tax lists or anything like that, you have any questions, we're here to help.
Yeah, I'd love to talk to you. Thank you all for joining us. We really appreciate you participating in this thing. I know a lot of you have no reason to trust the state, but we really appreciate that you're taking a chance and latching onto this thing. We hope that the payoff will be as great for you as it is for us. So can't thank you enough. Everybody have a great day.